Welcome to the SB Grid YouTube channel. Software tutorials by developers, lectures by structural biologists, unique content brought to you by SB Grid.
Welcome to the SB Grid webinar for April. This is Jason Key in Boston. Thank you for joining. A couple of announcements. We've got two more webinars going for this series. This is next month is Careless from Duca Hexter's lab at Harvard. So that's a application for sort of a next-gen X-ray data scaling application, which does really sophisticated sort of Bayesian scaling and can handle things like, stills from or Lowy data or all sorts of different X-ray scaling issues, and just plain old monochromatic data as well. So I'm looking forward to hearing about that. So be sure to catch that one. And then next month we have Pokey from Woong-Hee Lee, who is in Denver. And Pokey is sort of a next-gen NMR data analysis tool. So NMR Sparky has sort of run its course, and I think Pokey is sort of taking over where Sparky left off. So be sure to catch that one as well. With that, it is my pleasure to have Gustav Adritz joining us from right here in Boston. So he happens to be here doing his PhD at Harvard. He's going to be telling us about OpenFold. Retraining AlphaFold 2 yields new insights into its learning and mechanisms for capacity and generalization. As we get started, feel free to send questions in chat. You can save questions to the end. There'll be time. And with that, Gustav, go right ahead.
Yes, so hello everyone. As Jason said, I'm Gustav. I'm a first-year PhD student here at Harvard. And today I'm going to be talking about OpenFold, a project I worked on when I was a master's student at Columbia in the lab of Mohammed Al-Qureshi. So this is a bit of a Frankenstein of a talk in that it sort of covers the kind of basics of OpenFold. I've thrown in like a little workflow for inference and training to help you get started using OpenFold. And then at the end, just to give a flavor of the kinds of things you can do with OpenFold, I've included some of the results from a recent preprint from our OpenFold AlphaFold retraining experiments. So it may be a little bit all over the place, and I'm kind of testing this out for the first time here. But if for whatever reason something I say is confusing, lacks critical context, please just ask questions in the chat, or if it's urgent, over the Zoom. So I'll be starting with just a quick roadmap, to maybe establish some motivation for the project. I'll be talking about OpenFold and its features, and then, oh, that's weird. I definitely added a section here about the kind of tech walkthrough I'll be doing, just some basic inference and training commands. And then, as I mentioned, I'll kind of touch briefly on some of the things we found when we retrained OpenFold using AlphaFold. No, the other way around. When we retrained AlphaFold using OpenFold. Someday I'll get that right. So, as you all probably know better than I do, I'm concerned with the protein folding problem, which is the question of transforming a one-dimensional protein string and its associated metadata, like a multiple sequence alignment and a template, into a prediction of the three-dimensional protein structure. And the reason we care about this is, of course, that the protein's three-dimensional structure largely determines the protein's function. And so this is an important question for applications like drug design, or very many general questions in structural biology. So they've been trying to do this computationally for a very long time, but until quite recently, in 2016, the accuracy of the best computational methods was still in excess of 10 RMSD, which is very far from experimental accuracy, represented approximately by this red line down here. In 2018, as we probably all know, DeepMind entered the fray with AlphaFold. But the first AlphaFold was very much just kind of a refinement of existing techniques. They engineered it better. They added more compute. They used more recent neural network architectures. But it wasn't really a radical break with the past in the same way that AlphaFold 2 was. And indeed, AlphaFold 2 completely, well, it's been said that AlphaFold 2 solved the protein structure prediction problem. Of course, in the 2 years since, or 3 years since it came out, we've moderated those claims a little bit, but there is something to be said about just how much better AlphaFold 2 was than prior techniques, and also how innovative and how original the AlphaFold 2 architecture is. They redesigned it from the ground up using a whole bunch of completely original ideas that apparently, just given the ablations in the original paper, contributed a lot to the kind of remarkable success of the architecture. So one of the remarkable things about AlphaFold 2 is not just that it can predict monomeric protein structures with high accuracy. It's also been successfully adapted to a whole bunch of different domains as just like a quote unquote foundation model in structural biology, right? Without any further retraining. You can kind of hack AlphaFold to do peptide protein docking. You can hack AlphaFold to do multimeric structure prediction without any kind of retraining. Of course, since the original AlphaFold 2 came out, DeepMind's released AlphaFold Multima, which was explicitly trained to predict multimeric protein structure. But before then, you could do it just by kind of adding, like hacking the residue and the like positional encodings of the model. You can also use the kind of confidence module in AlphaFold to rank candidate protein structures. This would work out of Harvard. So really, it's a remarkably powerful model that can do a whole bunch of different things, like way out of its training distribution. And indeed, the model itself was only ever trained on small crops of proteins of length up to 384. Even if you train on crops of length 256, the model does remarkably well. So there's something magic about it. It knows much more than previous models of its kind. I'll be focusing just for a minute on AlphaFold Gap here, because I think it tells a nice story for why we decided to make OpenFold. So soon after the release of AlphaFold, in what may be one of the most cited tweets of all time, Ming-Kyung Baek pointed out that you could just add a small gap to the residue indices in AlphaFold 2 and trick it into predicting multimeric protein structures with relatively high accuracy. And this was one of a series of like related techniques. You can also add like a long glycine linker between the two chains to predict, like kind of fool AlphaFold into predicting them as a multimeric protein structure. But yeah. So for a while, these started to like outperform the existing like state of the art for protein docking. Here, AlphaFold Linker and AlphaFold Gap. Very soon thereafter, prior techniques were kind of upgraded with AlphaFold, and that kind of gave you a small bump as well. But none of these things could really compete with the DeepMind itself, right? Which used the AlphaFold training code, which had until this point had not been released, and simply like naively retrained AlphaFold 2 on multimeric chains, on multimeric proteins rather. And just by doing that, they were able to kind of completely outperform all of these kind of hacky approaches that kind of use the monomeric AlphaFold 2 for this purpose. And then at CASP 15 this past year, you'll see a similar story. Here, like most of the like top and the entries were just kind of like refinements of AlphaFold 2 that kind of upgraded the, for example, sampling procedure. One entry just kind of enabled dropout during inference, for example, sampled a whole bunch of protein structures, and then used AlphaFold 2's remarkably powerful confidence metric to choose the best one. This is remarkably effective. Other techniques kind of embedded AlphaFold 2 into like kind of prior protein folding pipelines, like ITASR. They added like better MSA computation and so on. And like by doing this, they were able to kind of gain a small edge over kind of vanilla AlphaFold 2, represented here by CoLabFold and here in green by just the original repository. CoLabFold, of course, has they use MMSeqs, and it's a related technique. But despite all of this, at CASP 15, the organizers announced that they had asked DeepMind for an unofficial kind of baseline. And what DeepMind did was they just kind of took AlphaFold 2, they added all of the chains that had been released since AlphaFold 2's cutoff dates, the original cutoff dates in 2018, retrained it, like used a longer crop length and so on, just small modifications to the original architecture. And by doing so, they were able to match the performance of kind of the best techniques produced by all of this painstaking effort at CASP 15. None of which actually involve significant retraining or like fine-tuning of the original AlphaFold architecture. This top entry at PESI Foldings did like some the fine-tune AlphaFold a little bit, and they were, by this metric, the top entry, right? So I think overall the AlphaFold gap story and now this shows the kind of remarkable power of the ability to retrain or fine-tune the architecture, right? There's only so much you can do with the inference code alone, which is what, in fact, DeepMind released to the public in 2021. So another thing I'd like to note is that, as I mentioned, AlphaFold 2 is a remarkably kind of complex architecture. GPT-2 and GPT-3 differ by a couple of like hyperparameters in a config file, right? AlphaFold 2 differs from AlphaFold 1 in like profound ways. It has this like 2-track transformer architecture. It has this kind of recycling procedure. It has a kind of novel structure module that differs from other approaches. And in fact, what people expected AlphaFold 2 to be in this kind of awkward period between CASP 14 and the release of the AlphaFold 2 paper in 2021. It has a whole bunch of weird like geometric attention mechanisms inspired by graph neural networks, this like triangle attention, triangle multiplication. Basically, it has a whole bunch of small components that could very plausibly become useful in other domains and other applications in structural biology, right? So I think there is demand for a kind of software suite that includes all of these different components in a programming, like in the machine learning architecture that people actually use, and that is sufficiently modular that you can just take these things and plug them into other applications, like stuff in natural language processing or RNA structure prediction or what have you. So that's why we made OpenFold. Just to summarize, one, we wanted to be able to retrain AlphaFold 2 and just to understand its properties. This was not possible with the DeepMind code release, which again only included the inference code and parameters initially under a very restrictive license that prevented you from using it in commercial applications. We wanted to be able to retrain AlphaFold 2 for new applications, and as I've been talking about for the past 10 minutes, retraining AlphaFold 2 is incredibly powerful and like kind of that beats out what you can do just by hacking together the inference pipeline. We wanted to remove barriers to training AlphaFold 2 style models by releasing a large database of kind of computationally expensive alignment data used to train the model. We wanted to release the parameters for AlphaFold 2 under a more permissive license than the one DeepMind initially used. In fact, this motivation was sort of obsoleted in January 2021. No, sorry, January 2022, after the release of OpenFold, when DeepMind changed the license on the original parameters. So in fact, you can actually now use the original AlphaFold 2 parameters in commercial applications, which is nice. And then, as I mentioned, we wanted to kind of make all of these new components available in efficient PyTorch efficient modular PyTorch code that you can just plug and play in other applications. And that's what we did. We created OpenFold, a open source reproduction of AlphaFold 2. It includes the entire inference pipeline, and we also reconstructed the training pipeline. And in fact, it is sufficiently faithful that you can take the OpenFold model, plug in the AlphaFold 2 weights without any modification, and obtain identical structures. This was important for us just because, even though it's kind of a nightmare to maintain, just because we wanted to establish confidence that OpenFold is in fact a reproduction of AlphaFold 2, and wanted to make sure that we didn't blow months worth of training on something that wasn't even the same thing as the model we were trying to reproduce. So I'll just preface this with a disclaimer that these next two slides are potentially out of date, just because between the time we created these plots and now, DeepMind released AlphaFold 2.3, a kind of updated version of AlphaFold Multima, which includes some optimizations to the training pipeline. So take this with a grain of salt. But basically, we spent a long time optimizing OpenFold to be faster, especially on shorter proteins below 1,000 residues than AlphaFold. Basically, this involved writing like custom CUDA kernels. We added like a dynamic chunk size selection mechanism. So OpenFold will actually scale up the chunk size used during inference, unlike AlphaFold 2. We have like torch script tracing. This is also a little bit out of date in that since I made this plot, Torch has also released Torch 2.0, which includes like a kind of compilation facility that kind of matches what AlphaFold 2 has in JAX. So we are optimistic that we can make OpenFold even faster using these new tools. And then similarly, we also made OpenFold a lot more memory efficient than AlphaFold 2. AlphaFold 2 caps out on the A100 we used 40 gigabytes at around 2,500 residues. Because of some, again, custom CUDA kernels we wrote, we added some like offloading, we did a whole bunch of careful in-place operations. You can actually run OpenFold on very, very long chains almost longer than the kind of the AlphaFold / OpenFold weights can actually handle. So at this point, the structure is not particularly coherent, but at least you can run them. And these figures are all from a single A100 40 gigabyte GPU. For those of you who are actually kind of actively using OpenFold, you can actually activate the kind of the very, very conservative mode using this long sequence inference flag. I'll mention that these optimizations that we made for the speed and also memory are not exactly orthogonal. And as you can see, the runtimes for these like kind of really, really long proteins are quite large. What's nice about OpenFold though is that you can kind of like pick and choose which optimizations you actually want and kind of continuously vary between kind of fast inference for short proteins and like slow memory, like conservative inference for very long proteins. So somewhere on the spectrum is a nice middle ground for your application. We used OpenFold to retrain the model from scratch, producing a suite of independent parameters and released them to the public under an open license back in, I think, June of last year. And we managed to match the accuracy of AlphaFold 2 on single chains. Basically, these are results from our held-in validation set. Excuse the resolution. I'm not really sure what happened here. But basically, I have GDTTS for AlphaFold 2 on the x-axis, OpenFold on the y-axis. As you can see, they're pretty much neck and neck, and they are in some sense complementary, right? So there are lots of proteins for which OpenFold does well and AlphaFold does poorly, and vice versa. So you could imagine running OpenFold and AlphaFold in an ensemble to kind of improve on both of them. And as I mentioned, you can actually run OpenFold with AlphaFold 2 weights, so you don't even have to download two different protein structure prediction pipelines to do that. We participated at CASP 15. Actually, I'll come back here. OpenFold is somewhere down here. We lagged behind the AlphaFold 2 baseline just because we were still training the model when CASP began. And then on top of that, we had some annoying bugs. We were like artificially limiting the depth of the MSA. So what I did here was I just took the single chain targets from CASP this year, CASP 15. I reran both OpenFold and AlphaFold. And again, it's the same story. OpenFold and AlphaFold are pretty much neck and neck. There have been some questions about OpenFold's template pipeline. In fact, when we trained OpenFold back in June of last year, there was a bug in the template pipeline that caused the OpenFold weights to kind of disregard the kind of template sequence. Sergey Ovchinnikov has pointed this out in a bunch of Twitter threads if you're interested. But sorry, my phone is ringing. Basically, unless you're kind of like in a subtask that cares very much about templates, OpenFold and AlphaFold are pretty much neck and neck. And the nice thing about OpenFold is that even if you do encounter issues with your particular application, you can just fine-tune it or potentially even retrain it. As I'll mention later on in the talk, that's not as burdensome as it sounds. So on top of that, we actually released the entire training set we use for OpenFold. Basically, we computed 15 million Uniclust 30 MSAs in the manner of AlphaFold 2. And then on top of that, a whole bunch of MSAs for the PDB proteins using more diverse data sets. We then took these 15 million and then filtered them down to a set of about 5 million, maximizing diversity and MSA depth and so on, and then released them under an open license on Rhoda, the registry of open data on AWS. So these are available for download and, can be used for either training OpenFold, fine-tuning OpenFold, or other structural biology tasks of your choosing. This includes, by the way, templates. We added backups for the TIFF files we use because those are always changing. So yeah, this is hopefully our goal was to make this like as plug and play as possible. We want you to be able to download this stuff and kind of reproduce our own reproduction experiments without too much hassle. We offer pretty diverse inference and training modes. OpenFold training supports deep speed. We have PyTorch Lightning integration, so you have access to the whole range of functionalities that entails. We have a Docker container, and then we also actually added an FP16 training mode, something that's not, well, the original AlphaFold 2 code doesn't include any training modes at all, but this actually required some engineering effort. So you can now train in FP16 if you have an older GPU, and bfloat16 like the original AlphaFold 2 if you have an A class GPU. Ampere, is that what it's called? Anyway. Yeah, so a range of options. You can even do full precision training if you like. We actually, so as I mentioned in the first point here, we erred on the side of kind of reproducing AlphaFold as closely as possible. So in general, we were extremely conservative with changes we made to the architecture. Nevertheless, we were able to make some changes that kind of minimally affect the output activations, such that you can still run OpenFold with AlphaFold 2 weights, but kind of improve the experience a little bit. A bunch of those things involve kind of normalizing large tensors. That was important for the FP16 training mode, which of course is a smaller numerical range than bfloat16. But we also changed the training procedure a little bit. Specifically, we found when we retrained AlphaFold that it was quite unstable. Very often you would end up, so like the default outcome was to end up here on this high plateau at around like 0.82 LDBT. This is just training on PDB chains without the distillation set at the moment. And you actually usually very quickly end up on this plateau. In a large fraction of cases though, in these experiments, it was 60% of cases, you actually ended up on this much lower plateau of LDBT. And only later, after training for thousands of steps, would you actually end up on at the level of these successful runs. So I'm looking at the blue plots now. This is kind of vanilla AlphaFold or vanilla OpenFold in the model of AlphaFold 2. And sometimes you either wouldn't reach this higher plateau by step 10,000 or you would never reach it, right? Like at this point, like AlphaFold 2, the iteration times are like 7 seconds per step. And so with gradient accumulation, which we were using here, this represents a huge amount of compute wasted on a run that like doesn't really seem to be converging anytime soon. What we did was we found that the cause of this instability was that well, maybe not the cause, but we found a workaround. We found that in the early phases of AlphaFold 2 training, this like clipping procedure that AlphaFold 2 uses for FAPE, the main structural loss, was too aggressive and was in fact kind of nullifying most of the relevant training signal in the kind of unstable early phase of training. So just by clipping a small fraction of every batch instead of a large fraction of batches, sorry, we were clipping a large fraction of each batch rather than completely clipping a large fraction of batches, we were able to like completely stabilize AlphaFold 2 training. And now I think this is like 20 runs using the new technique. You pretty much always converge to the higher plateau and actually quite a bit faster than the original AlphaFold 2, about 30% faster. So that's nice. We support AlphaFold gap inference at the moment, that kind of hack I mentioned. So you can actually predict multimeric structures with OpenFold, but we actually have a proper OpenFold Multima implementation coming soon. Christina, a PhD student in Mohammed's lab, has been working on this, and it's quite close to completion. So soon you'll be able to run OpenFold with the AlphaFold Multima weights as well. And then hopefully also train the your own Multima models using OpenFold Multima. And then on top of that, basically OpenFold is quite a large community now, implementing a whole bunch of new features. ESM2 used OpenFold as part of its code base and has been kind of pushing PLs to the OpenFold repo. That was in a recent paper in Nature Biotechnology that used OpenFold. There's lots of efforts like FastFold, Unifold based on OpenFold that kind of improve it in various ways. This one claims to reduce the OpenFold training times from 11 days to 67 hours. So that's out there, open source. And then on top of that, we also have constantly new PRs with nice little features added all the time. So it's an evolving repository, and we're excited to see all of the cool things that people are able to come up with. So that's OpenFold. I'm now going to be walking through simple use cases like predicting a single and the protein structure, and then also like training OpenFold from scratch. I realize that kind of the talk is not necessarily the best format for like a software demo, but hopefully since it's being recorded, you can kind of return to this later on and kind of walk through this in your own time as you use OpenFold. I'll mention too that as I mentioned, we have like a whole bunch of different features, like these like various long and the sequence inference modes, these like fast inference modes and so on. I won't be touching on all of that here, but hopefully the documentation is good enough that you can kind of piece that together. I'll be kind of mentioning things here and there, but this is not the full story. And I recommend looking at the README if you actually intend to do this yourself. So the first thing you'll want to do is go into the scripts directory. We actually have a whole bunch of nice utilities for like computing MSAs, for downloading the databases, for installing the software and so on. There's a whole bunch of nice stuff in there, including a script that just installs everything for you. This will download the AlphaFold 2 parameters, the OpenFold parameters, all the software you need. It will download the smaller databases, I think, the utilities that you need to download the databases. Basically, hopefully you just be able to run this and get your environment set up in a single step. Next, oh, by the way, this is the inference pipeline for now, even though you do need to do this for both inference and training. This is next. If you intend to compute MSAs on the fly, you'll want to either download the AlphaFold 2 databases from DeepMind's mirrors, or download databases that ColabFold uses. We actually support either kind of HHBlitz MSA computation like AlphaFold does, or MMSeq's MSA computation like ColabFold does, and these scripts will download the right databases for either of those two use cases. MMSeq is of course much faster and doesn't really have a noticeable downside compared to the original and the MSA computation procedure. But we include this just because, you know, for completeness' sake, this is what AlphaFold 2 did, and so we want to kind of make that option available as to ensure that the reproduction is as close as possible. Next, this is not the best slide I've ever made, but basically in the OpenFold directory of the OpenFold directory, there's this file called config. That's where the model config lives. So if you want to, for example, change the chunk size or activate all of these like different inference time optimizations, you do that right there in the config file. Next, you just run the inference command. Basically, all of the OpenFold scripts, including the training scripts and the inference scripts, accept as, okay, well, I shouldn't lump together the training and the inference scripts, actually. For the inference scripts, it expects a directory full of FASTA files, so just one directory filled with FASTA files with unique names, a directory of MMCIF files used for templates, so that's where the templates come from, and then a whole bunch of paths to various alignment databases, right? If you just use this download AlphaFold DB script, you can probably just copy and paste this directly, right? There's UniRef, there's Magnify, there's PDB70, there's all the like binary paths to the various kind of alignment utilities you'll be using, and then the config presets that you want to use. As I mentioned, this will accept both OpenFold checkpoint paths from our own weights or AlphaFold 2 weights in NPZ formats. So you can just take the JAX weights and plug them right in. Anything worth mentioning here? Oh yeah, of course. I've highlighted this flag down here. If you have MSAs computed already for your sequences, which if you're running it more than one time is likely to be the case, you can just use this use pre-computed alignments flag to pass to the directory of MSAs. I'll talk about the expected format of that directory when we talk about training, but there's no need to actually run the entire kind of expensive MSA computation pipeline every single time if you already have these MSAs pre-computed. So that's it pretty much. If you want to, you can also run if you put multiple chains in your FASTA files, by default, OpenFold will actually predict them using AlphaFold Gap as a multimer. So that doesn't require any additional tinkering. You just you can mix like monomeric sequences and multimeric sequences, and the script will just handle it. So, I mean, I'm going to say now it's as simple as that, but of course nothing is this simple. And if you try this and it doesn't work, we have an issues page that you can use, and a longer README that maybe includes details that I forgot to mention here. So for training, it's quite similar. Initially, you'll want to download the same databases, either the ALPHOLD databases or the MMSeqs databases, and then run one of precomputealignments.py, again in the scripts directory, or precompute alignments MMSeqs, which we'll use MMSeqs instead. Here, you'll want to give it a directory with MMCF files for templates, the alignment directory. And actually, the MMCF directory here contains also the sequences that you want to precompute alignments for, right? The alignment directory, which contains the output directory, that's where it's going to actually dump the precomputed alignments. And then again, all of the different databases and alignment tools you want to use. I'll mention here that the MMCF directory, despite its name, also accepts PDB files and also .core files from Mohammed's protein net. So you can just mix and match. Pretty much, we support a number of file formats. Odds are that if you have a protein structure file, you can just dump them in the MMCF directory, and OpenFold will know what to do with it. Yep, as I mentioned, you can also run MMSeqs. That's going to be faster and almost probably just as good. So this is the promised structure of the MMCF directories used for pre-computed alignments. And this is actually the format also output by these pre-computation scripts. Basically, all you want is a... Oh, I'm sorry. This should not say MMCF tier. This should say alignment tier. My bad. In any case, all you need is a directory of directories where the directory names, at the middle layer, correspond to the names of structure files in your MMCF tier. So if you have like 7sbq.sif in your MMCF tier, you'll want 7sbq here, or 7sbq_a for the first chain or whatever. And OpenFold will kind of match those two things up. And then inside the alignment directory, you can have alignments in pretty much any format. You can just throw in A3Ms and like HHR template hit files and so on. And OpenFold will combine them into inputs to the model. And so there's no need for these specific files that I've listed here. And this is just what's output by default by the alignment script. So next, because parsing MMCF files is so slow, you'll want to generate 2 caches that will just speed things up a little bit during training. So basically, you just pass it your MMCF tier and the desired output file, and it's multi-threaded. So if you have CPUs, you can go wild. This one speeds up template filtering, and then this one speeds up a whole bunch of like chain filters during training. It will, like, OpenFold has a whole bunch of like stochastic filters that will filter things by like cluster size and so on. This just takes care of that. I've been meaning to combine these two. It's a little bit awkward to run to generate 2 different caches based on the same data, but for now, at least they're separate. And then I'll mention actually that if you want to just skip this entire process, not have to compute any alignments, and you just want to use the MSAs that we've released, all you need to do is download them from AWS. This includes everything, including the distillation MSAs that we released from ALPHOLD 2's distillation sets. And then this script file just changes the format into the format right here expected by OpenFold. By default, the Rota format is a little bit different, just so you can download subsections of the data without. But yeah, so this is what you do to prepare the kind of Rota data for OpenFold training. I'll mention that if you're training on a very, very large number of chains, this format can become quite cumbersome. Like, I think most cluster file systems don't like directories with like hundreds of thousands of subdirectories. So what you can do is, once you've computed your alignment directory or just partial alignment directories, you can actually compress them into these like kind of database files that we have in the scripts directory. Again, this is in retrospect not the world's greatest slide. But yeah, these can be found in the scripts directory of the OpenFold repo. And this basically just allows you to condense a whole bunch of alignment directories into a smaller number of files that can be read by OpenFold. And you can actually specify the degree of sharding occurs if you have a maximum file size or something. So this is recommended for kind of real training jobs. And then finally, you just run the training script. You have your MMCF directory with your, like, again, it's misleadingly named. This could be any kind of like structure file, PDB, MSCF, .core, whatever, what have you. You have your alignment directory with your pre-computed alignments, including MSAs and templates. You have a template MMCF directory, which could be the same as your the target MMCF directory. And then you have just a directory where it dumps like checkpoints and outputs and logs and so on. Anything worth mentioning here? So the training script supports multi-node, multi-GPU training, thanks to PyTorch Lightning. So that's relatively easy to do. It supports deep speed, so you can just pass it a deep speed config right here with your kind of optimizer settings, your zero optimizer settings. You can resume from an OpenFold or an AlphaFold2 checkpoint if you want to fine tune it. You can you pass your two caches here. This is the template cache I mentioned. This is the training cache I mentioned. So those are generated by those two scripts I mentioned earlier. And then this obsolete PDBs file is just, it comes with the MMCF files that you download from PDB. And that's pretty much it. This will start an OpenFold training run and get you started. So this is kind of the weird, awkward Frankenstein juncture in the talk that I mentioned, where I'm going to start talking about things we actually found when we did this ourselves, just to give some flavor of what you can actually do with OpenFold. I'm wondering, is this a good time to maybe pause for like technical questions that might become less fresh after the maybe extraneous results section?
Technical questions? I think we had a couple, but I think as you went along, you answered, you can specify the location of the cache file and, those things for like, if you're working in a high performance computing environment, that's important. You don't want to necessarily. Right. And if anyone has some technical questions, you could send them by chat. Otherwise, we can continue and I can interrupt you if people send them. One that came up is that, oh, here's one. Could you say a little about using experimental quality scores in model training?
Experimental quality scores in model training. So, I mean, I'll scroll actually back to the architecture. So you're talking about using the quality scores as like an input to the model, basically, to train some variants of AlphaFold2. Just to clarify.
I guess I'm thinking for the basis of this question is that a structure in the PDB also has some experimental data that supports that model.
I see. Yeah,
some of them it's very good, and some of them it is less good just because the experimental data is limited. And it seems like that would be a fruitful thing to include in this process.
Yeah. So, but by default, that's not included. But right here in the kind of embedding section, there's like an embedding head with a whole bunch of different components. There's a recycling embedding head, there's the NSA embedding head, there's the template embedding head. You could very easily add like kind of experimental data embedding head and just mix that in, right? So that's the kind of thing we actually hope to kind of enable with OpenFold. Hopefully it should be fairly straightforward.
Thank you.
So if that's it for the technical questions, I'll keep moving, but again, feel free to interrupt me.
Yep, go right ahead.
So basically, one of the goals of OpenFold, as I mentioned, was not just to make this kind of available to the community, but also to understand facts about the AlphaFold2 training process that kind of went unanswered by DeepMind in the original paper. DeepMind is the supplement to the AlphaFold2 paper is one of the most beautiful documents I've ever read in the ML literature. It's so long, it's so detailed. It actually kind of made OpenFold possible because they kind of described the methods in sufficient detail that we were able to reproduce them. And they also have a whole bunch of nice ablation studies and so on, the effect of recycling over time for different proteins. But they actually didn't answer many questions about what happens during training. For example, we had many questions about how much data do you need to train AlphaFold? How quickly does it converge? Like what the intermediate proteins during training look like? Does a protein from early on in training look like a protein from early on in the recycling process? Like these are things that kind of we wanted to know, and we were able to kind of like begin to answer these questions actually using this retraining run that we did with OpenFold. So one of the most surprising findings was that OpenFold and AlphaFold, presumably AlphaFold2 as well, converges incredibly quickly, especially if you have the kind of fixed the training and the procedure that we implemented here. So in just like 2,000 or 3,000 steps, representing like about 2,000 or 3,000 A100 hours in total, you can actually achieve like 95% of OpenFold's final accuracy, which actually opens up a whole bunch of nice possibilities for kind of retraining OpenFold on the using limited computation, right? For example, in academic settings, right? As you continue to train the model, it will improve in various subtle ways, like for various obscure secondary structure elements, it will continue to improve slowly. The violations kind of slowly improve over time, but really like this model early on training is in like a really sweet spot. This like completely blows prior protein structure prediction methods out of the water. AlphaFold2 is nowhere near 0.8, 2 or 3 PLDDT. And in fact, this model, which is trained with the distillation set as well, it goes even higher. I think at this point it's around 0.87 or 0.88 LDDT compared to the final value of 0.90, right? So truly, you can kind of get away with training AlphaFold2 for very, very little time. And in green here, so the AlphaFold2 model is trained in a number of stages. There's the initial training phase, which is just smaller crops to achieve like kind of faster throughput. And then a slower fine-tuning phase, which activates a violation loss, which increases the crop size and so on. And while this is important for kind of reducing violations, because as I mentioned, the initial training phase doesn't actually have a violation loss at all, and you will actually see pretty unsightly clashes and so on at this point. The fine-tuning stage does resolve that a little bit, but it doesn't markedly improve the accuracy of the model, even if you evaluate it on longer chains, right? So this is actually great news, right? Because the fine-tuning stage, this little green section here, cost as much compute as the entire orange section. So basically, you don't need as much compute as we thought to train AlphaFold2 style models, which is great. We also found that the AlphaFold2, again, the resolution is not great, but hopefully still visible. We also found that the confidence metric of AlphaFold2, which has been like one of the most remarkable features of the model, how well it can assess the quality of its own predictions, even in cases when it fails catastrophically. This capability develops extremely early on in training. So what I have here is PLDDT, the predicted confidence of the model, versus LDDT, the actual quality of the model, the actual accuracy of the model. This is a per residue score averaged across validation sets across time. So the color here represents the training step. And 5,000 here, as I mentioned, is extremely early on in the training process. It's right around here, right? So very, very early on, even by step like 1,000, it can pretty accurately assess its own quality, right? Which is, again, great news for training this model on new data, right? Like you'll have a pretty good idea very early on how good your model is and how good you want to make it, right? So that's fairly remarkable, and I'm not aware of other models that are this good, this well calibrated, like this early on in training. As I mentioned, actually, let's skip this slide and go straight to this one. So what I have here is the kind of the F1 scores for the accuracy of a whole bunch of different secondary structure elements over time. And as I mentioned, very early on by step like 4,000 or certainly before step 5,000, it pretty much masters alpha helices and beta sheets, which kind of gets you almost all the way there. And then in a staggered fashion, it slowly starts to pick up on these more subtle secondary structure elements, which is nice. So it learns the most important material first, and then only later does it kind of fill in the gaps, which makes it possible again to do these abridged training runs on very little compute. And just kind of breezing through this, we found that the model learns on multiple scales. So basically what I have here is GDT and DRMSD scores for different crops of validation proteins of various lengths. So the blue one here is the model's accuracy over time on small crops of about 10 residues, exactly 10 residues drawn from these training proteins. And then we have 20, 50, and the entire proteins. And as you can see, it kind of masters local structure first, and then slowly kind of grows outward, improving second in the intermediate and the size crops, and then larger ones, and finally it completes the full proteins. Again, I want to stress that this is still before step 5,000. So it even though it happens in this staggered fashion, very early on, you end up with very high quality structures. The same story holds for helices and sheets. Longer helices are kind of learned slower than shorter helices. Bigger sheets, represented here by the number of threads, are learned slower than, oh, sorry, these are the sheets over here. I was pointing to the wrong one. Larger sheets are learned slower than smaller sheets. So there's a very kind of orderly progression in the quality of protein structures predicted by OpenFold over time. We found this like nice little detail that hopefully this works. The videos usually are a bit spotty. Oh no, it took me to a browser window. Okay, I'll use the static slide instead. That was an animation. But basically what happens is that as you train the model, protein structures undergo this kind of progression of dimensionality. So initially it kind of masters the first dimension of the protein structure, producing these like kind of quote unquote one-dimensional logs in three-dimensional space. And if you actually look at the quality of the prediction on these logs, it's actually quite good. So the residues are ordered roughly as if they were like kind of projected onto this one-dimensional log from the kind of final three-dimensional structure. So they're in approximately the right order. Next, the log kind of expands in the second dimension to produce these like kind of flat but still like largely accurate structures. And then very, very quickly thereafter, kind of blows up in the third dimension, becoming like a pretty accurate prediction of the final structure. And only then does it start to fill in the secondary structure elements to the point where it's kind of recognizable by PyMol here. So it's almost as if it's learning like subsequent PCA projections of the final protein structures, which we thought was pretty interesting and pretty unexpected. So yeah, these are just different ways of visualizing the same thing. Basically, there's what I'm visualizing here is the kind of movement of residues in the directions of the kind of principal eigenvectors of the final structure, right? So initially, there's a lot of movement in this kind of like most significant direction. Then there's a lot of movement in the kind of next significant direction in the kind of two-dimensional phase, and then finally kind of fleshes out the final dimension as residues move in the third direction. And this is kind of like remarkably consistent across like hundreds of proteins that we test in. So it's pretty cool. And the reason this is cool is that like prior protein structure prediction methods would not have allowed this kind of progression. Like, for example, AlphaFold1 initial samples proteins initially from kind of a learned distribution over proteins, and as it folds the proteins by minimizing the potential function that it learns, it enforces like chemical constraints, which are like part of the potential, right? Of course, a completely flat two-dimensional structure like the red one here, predicted by AlphaFold2 in its intermediate stages, would have been completely impossible if AlphaFold2 had enforced these like strict chemical constraints from the very beginning of training. Because AlphaFold2 defers that to the fine-tuning phase later on, though, it's able to kind of take this unique path through the optimization landscape that potentially plays a role in its success. And you'd have to imagine that it does, right? Given how consistent this is for a whole bunch of different proteins and across training runs as well. So yeah, a nice little detail that we were able to find. I think the most surprising outcome though was that you don't really need data to train AlphaFold2. Like it's remarkably powerful at generalizing to unseen folds. So what I have here is basically the original OpenFold training run in pink here. So this is OpenFold trained on all PDB proteins. Note that we exclude the distillation set here. And we are also training with and without templates. As you can see, the templates don't really make a huge impact on the model's performance. What we find is that if you just uniformly sample proteins from the PDB training set, reducing it in size by a factor of like up to 100, it doesn't really affect the accuracy of the final model that much, right? Like even if you train on just 12% of PDB structures, sampled pretty much uniformly at random, you still converge to pretty much the same accuracy as the original model. And this holds pretty much down to 1% of the original training data. At this point, it does take a large hit, right? This is now down to like 67, I think, 68 LDDT. But I'll point out that this is still better than AlphaFold1 was on these proteins, right? So there's something magical about AlphaFold2. It really generalizes remarkably successfully to unseen folds, even if you kind of massively restrict its training data. Additionally, we were trying to break it just by continually reducing the size of the training data it was trained on, but we couldn't do it. So what we did was we started to filter the training data, not just uniformly, but like kind of adversarially. We took Cath classifications of proteins, Cath, of course, being a kind of the taxonomy of protein structure, with 4 layers: the topology, there's homologous sequences, topology, architecture, and class. What we did was we started moving up the Cath hierarchy, and instead of just like uniformly removing chains, we started removing entire like kind of classes and the categories from Cath, right? So what we did here was, again, pink is the default OpenFold training run on a validation set composed of unseen topologies. And what we did was we started like removing the topologies from the training sets. So the kind of orange one here is OpenFold trained on 50% of available topologies. The green one is 10%. The blue one is 5%. And again, like even if you, and these are all being evaluated on completely unseen topologies by any of the models, right? So even if you remove half of the available topologies, you barely make a dent in its prediction accuracy on unseen topologies. And if you train on just 5% of the available topologies, you still achieve like a pretty respectable protein structure prediction model, right? This holds if you go up to architecture. I'll note that the 5% one here is just a single architecture. So this blue one was trained on proteins from just one architecture, and it nevertheless achieves, oh, what happens to my y-axis? Okay, well, it almost reaches 0.6 LDC on proteins from an unseen architecture. No, not just one unseen architecture, from completely unseen architectures. Each chain is from 5 different unseen architectures. And again, the same exact thing holds for class. If you remove all classes but one, and then evaluate on class 3 proteins, which have both beta sheets and alpha helices, the model barely cares, right? Like the kind of green one here is trained exclusively on class 1 proteins, so mostly alpha proteins. The red one is trained exclusively on mostly base proteins, and nevertheless, they do pretty much just fine. And at this point, we ran out of ways to break the training set, like AlphaFold 2.1, basically. I'll note that these models do have some like strange like pathologies, right? Like, for example, what I have here is the ground truth for 2 proteins in orange here. This one being kind of a mostly alpha protein, this one being a mostly beta protein. And then I have the mostly alpha model, this kind of green or yellow one here in the second column, and then the mostly beta model trained exclusively on mostly beta proteins in red. And as you can see, the mostly alpha model is pretty good at predicting mostly alpha and the chains, and vice versa, right? The mostly beta one is pretty good at predicting mostly beta chains. But nevertheless, they don't really catastrophically fail on proteins from the other class, right? Like this one doesn't really get the exact fine structure of the beta sheets right, but the global structure is still like pretty much correct. It also hallucinates this alpha helix here, unsurprisingly. In the same fashion, the beta model will hallucinate like a little beta sheet here that shouldn't be there. But the global structure is still quite good. And these are actually like cherry picked to kind of maximize to kind of show the characteristics of these 2 models. In fact, if you evaluate these models across the validation set on mostly alpha helices, mostly beta, and on mostly alpha proteins and mostly beta proteins, the gap between them is not this large. It's only like 7 or 8 points on average. So these are kind of like perfectly functional protein folding models, despite their like massively restricted training sets. So I think I'll skip this slide just because I'm running out of time. So basically, OpenFold has become kind of the foundation of the kind of became the impetus for the Open Molecular Software Foundation, which is kind of a new academic industry initiative interested in producing kind of open source software for use on various structural biology tasks, like single sequence structure prediction, RNA structure prediction, DNA structure prediction. That they're interested in a whole bunch of different things. So yeah, we're excited about that. Everything that I mentioned today is also available in the preprint we put out back in November. So yeah, if you're interested, there's detail on all this stuff and more there. And finally, I'd like to thank both Mohammed, who kind of led the lab where this work was done, and then Nazim, who was the kind of co-first author on the paper, who kind of advised and kind of conceived of this project and deserve some credit here. So yeah, that's what I have.
Oh, fantastic, Gustav. Thank you very much. It's that training. It's fascinating.
Yes, it's awesome stuff. Yeah.
That's unbelievable. So I saw for questions, we've got you can hit the reactions button, and I think you can raise your hand, and it will pop you up to the top of the list. I believe there's a raise hand. Yeah, it's the big button right when you. I'm gonna raise my hand, see, and I think guests can go ahead and raise their hands. We're gonna do the raise the hands now instead of unmuting everyone, just because with this many people, there may be a few lurkers in there who would disrupt. But the you talked a little bit about the training process and the secondary structure elements, and you said that like it takes a while to pick up some of these like more rare things like polypropylene and like
Right,
and
right.
so I was like when you showed me that I was sort of like, oh yeah, that makes sense. Those are like not represented as frequently, so it probably takes a while to encounter them. They're not as but then on the next slide, the how quickly it picks up the secondary structure of just alpha and beta is like it does the shorter first, but like that's not like it's not like short helices are more common than like
No,
long
absolutely not.
helices.
So it really has
Right.
this kind of like locality and the prior, right? But probably because it's trained on these like tiny little crops of proteins, right? So it's sort of in some sense forced to learn like local structure before it kind of slowly generalizes to larger proteins as it kind of grows outward. Yeah. So yeah, we were actually quite surprised by how orderly this all ended up being.
Yeah, and I was wondering, you kind of hinted at this when you were like, you know, the ways you can break the training data set. Like if it never saw like some rare structural elements, like it never saw a 310 helix, or it never saw like a pi helix, or these kind of rare turns. Like, can it reproduce the things that it hasn't seen?
So I think our data is not conclusive on that question, just because there are still some beta sheets in the mostly alpha class, and there are still some helices in the mostly beta class, right? But like, just given how good it ends up getting at like pretty long alpha helix, this model is like barely ever seen an alpha helix, and it knows like pretty much exactly what to do, right? Like, I think there's reason to believe that it could just like kind of do these things that it's never seen before. And actually, what I'm saying, these architecture and topology splits are also, I think, quite telling, right? Because like this blue model here is trained on one architecture, like that's like very high in the CAP hierarchy, and it generalizes like really nicely to completely unseen architectures, which have structural motifs that it's really never seen before. Granted, of course, CAF classifications are like kind of quite global, right? And so if you have a model like sort of like masters local structure prediction before global structure prediction, you kind of expect it to be able to kind of hack together this kind of thing. But I think it's still remarkable just how little like diversity you need in the training data for it to kind of pick up on a whole bunch of things that it's never seen before.
Which would strongly suggest that given the size of the training set that it's seen, that and how often as we determine experimental structures, we keep finding the same folds over and over again.
Yeah.
How rarely we encounter new folds, that is. It's probably pretty close to being able to predict everything nature can throw at it, I would say.
I'd think so, yeah. And even if it can't, there's good reason to believe that it knows that it can't, right? Because the confidence metric like completely outstrips the kind of structure prediction metric. It's really pretty remarkable.
Wow. Any other questions? You can put them in the chat, or you can raise your hand. Anybody? Let's see. All right. Well, yeah, it's amazing to see how this all comes together. It's been, obviously a huge topic of interest. We've had it in SPGrid a long time. People have been using this for a long time, but this training piece, like how it came to be and how has really been a black box, for lack of a better term. So this shines an important light on like how all these things come together, like how sensitive this all is. I've seen people working on nucleic acids versions of these. Any insight into like if OpenFold or these metrics could be used for things like RNA?
I hope so. Like as I mentioned, like many of the components of AlphaFold2 are quite general, right? Like most of them are just basically like inspired by this graph neural networks, right? So you could definitely imagine using like triangle attention, like IPA for and sorry, my phone keeps drifting. Funny kind of like the molecular modeling and so on. And then I think separately from that, just the kind of generality of the components, I think the kind of data experiments that we did are pretty encouraging, right? Because like, of course, for tasks like DNA structure prediction, RNA structure prediction, you don't have as much training data as you do for protein structure prediction. And so if AlphaFold2's ability to kind of generalize from almost nothing carries over into these kind of like data sparse regimes, I think there's some hope that you could kind of achieve a like pretty respectable model even without kind of the huge amounts of training data available to models like AlphaFold2.
It hints at being able to train on some chemical space as well, right? So the sort of vast chemical space that you'd have in like drug discovery. I mean, it's almost like, universal size, you may be able to, within some limits, train on.
Yeah, potentially.
Yeah.
And also, like, these are all from scratch, right? So you could also imagine doing like kind of multimodal learning, fine-tuning weights like this. Like this is just really like scratching the surface.
Yeah, that's fantastic. Well, if there aren't any other questions, oh wait, there's one more. No, that's an old one, sorry. So if there aren't any more questions, Christoph, thank you very much. This is
I actually see one question in the chat here about
really?
AlphaFold 2.3 weights.
Oh yeah,
so
you said something about it, but yes.
Yeah.
Go ahead.
So AlphaFold 2.3 was actually a multimer release. They didn't actually update the monomeric weights in AlphaFold 2.3. And at the moment, OpenFold only actually accepts the like monomeric weights. As I mentioned, though, we are implementing OpenFold multimer, and hopefully that should be out like in the not too distant future.
Oh, great. Thanks for catching that. That's an important distinction between the monomer and multimer weights. Great. Well, thank you very much. And we have OpenFold in the SB Grid collection. The multimer version is coming, so we'll be excited to add that one. And everyone, thank you for joining.
Thanks for having me.
Yep.